All right, so where our start is right here. 3 different kinds of muscle: cardiac, smooth, skeletal. This chapter is by far leaning more towards the study of skeletal muscle. We'll touch on a little bit of cardiac and smooth, but just a little, because you get more of that specifically in A&P 2. Right? Our job right now is more skeletal. So look at the orientation of the whole muscle itself. It's very similar to what we saw in the nervous system, in that what we saw is if I have about 5 or 6 markers in my hand here, right? It's a bundle of markers, you could say, right? If each marker represents a muscle fiber, then I have a bundle of muscle fibers, right? What we're going to call the muscle fiber is the muscle cell. So that's the first big thing you got to get into your brain. When I say muscle cell, I mean muscle fiber. And when I say muscle fiber, I'm talking about the muscle cell. What is every cell surrounded by? What's its barrier? Every cell. The cell membrane, the plasma membrane, right? So consider the plastic coating on this marker. It's cell membrane. But instead of saying a cell membrane, we call it a sarcolemma. So now every muscle fiber that I'm holding, every marker that I'm holding, has a sarcolemma, right? Because it's its own cell membrane. So when you think of muscle cells, you need to not be thinking about some nice round cell, right? This is more cylindrical. It's a tube, right, of a fiber. And inside of that cell is more fibers, smaller fibers that we'll see. More protein filaments is what we'll see. But for the moment, consider each marker to be a muscle fiber. When I've got a group of them, I call it a fascicle. And when I have several fascicles together, I call it the entire muscle. So in this picture on the board, the smallest thing sticking out says muscle fiber, right? It's one of about a dozen inside this one fascicle, and that one fascicle that's sticking out is one of about 7 or 8 making up the whole muscle. Can y'all see that? That's the big thing. There's a whole section of the test in matching where you're going to be asked, groups of muscle fibers are known as fascicles, right? Groups of fascicles are known as the entire muscle belly. Right. Or you could go the other way. The entire muscle belly is made up of fascicles. Each fascicle is made up of muscle fibers, right? Now, if we go inside each muscle fiber, we dive in, which you can't see on the picture, but we're going to do it mentally. If you dive into each muscle fiber is made up of myofibrils. So I've got a PowerPoint slide that explains all this, but I'm doing this first because it's one of the big things I need you to get. So each muscle fiber, if you had fibers, you can barely see them in this picture, but sticking out of that muscle fiber would be smaller protein fibers, which are called myofibrils. And then it goes even deeper than that. So that's where we're going in just a little bit. But let's do the layers that we can see on this picture here, because again, I've got another slide that explains all this, very detailed. What is that wrapping around the fascicle? Perimysium. What's that wrapping around the entire muscle? Epimysium. Right? So the epi is around the entire muscle, the peri is around the fascicle, just like we said for the nerves, right? We had perineurium around the fascicle of axons, remember that? And endoneurium around each axon, right? So now around each muscle fiber is going to be an endomysium that we're going to see. So that will be coming up. But because we only have one class period to cover this, I usually like to mention it twice. So I'm mentioning it now, and we're going to pretend in 20 minutes when I mention it again, you're going to say to yourself, oh yeah, I heard that before. So it's like we're living 2 days in one. Because typically I always cover this in 2 days, not one. All right, here's some of the general stuff that we have to know about muscles. General functions here: produce movement, maintain posture, stabilize joints. We've all been through that. Thermogenesis is nothing more than it produces heat. The muscles counting as a motor in the body, they produce motion. Anything that produces energy always lets off heat, right? So when we use our muscles, we give off heat. Other considerations would be, which is not in the textbook, surprisingly, or not surprisingly, is that muscle mass, skeletal muscle mass, is one of the prime indicators of absolute whole body health, meaning your immune system. If you have no muscle mass, your immune system is basically shot, for the most part, right? Those people that get older and lose all their muscle mass, their immune system drops dramatically. So immune function, right, is very much helpful with immune function. And also glucose management. Those people who have no muscle mass do not manage sugar properly, right? Okay, so I can go off of that for like hours. Main properties here. Excitability just means any muscle can be excited. It's responsive to stimuli. Contractility means when it gets excited, what's it going to do? Contract. Perfect. Extensibility means it has the property that can be stretched. So muscle fibers can be stretched. The next part is when they're stretched, they have the ability to do what? Return to their original tone, and that's the elasticity. So know the difference. Extensibility is stretching. Elasticity is returning from the stretch. What happens for people that have postural distortions, which they call elastic and plastic deformations, is that let's say you sleep on one side and you sleep on 3 pillows, so your neck is like this all night long, right, as you're laying on that high pillow. Or you fall asleep on your head like this on the couch and your neck is the same way. That will stretch out these muscles on one side, right? If you do that enough through months and years, you will clearly mess up that muscle where all it does to return to shape is a spasm. So that's how postural things can start off, simple deviations, but they turn into big muscle problems and spasms because they lose their sense of elasticity. And that's all over the place. All right, basic properties of these 3 muscles. For smooth muscle, we consider it to be non-striated, involuntary. This is old news, I think. We've done this before. So which one of these on the picture is smooth muscle? On the far right. Yeah, okay, good. Slow and sustained contractions. Once smooth muscle gets going, it's pretty powerful, but it's not a very fast contracting muscle. It is in the sense of the tone of the blood vessels. It can contract fairly fast because the surface area is small. But you don't want your smooth muscle in your colon contracting very fast, do you? No, we want it to be very slow and controlled, yes. I think we can all appreciate that. I swear I'm still 14. I can never give that up. Another name for a muscle cell, we got lots of names. We can call it a muscle fiber, we can call it a myocyte or a myofiber. All the same thing. Myocyte, myofiber, muscle fiber, or muscle cell. All the same thing, no matter what kind of muscle you're talking about. Next up, cardiac. Striated in appearance. It has the striations just like skeletal does. Still involuntary. Found only in the heart. And it's more branched. So cardiac muscle has lots of branching to it because they're all networked together. And what do we call these network links right here? These rigid lines, the zigzag lines that branch each muscle fiber to the next. Those are the intercalated discs right here. Intercalated disc. Those are those connections. So the cell junctions that we see are gap junctions and desmosomes. So what do you take when you take a gap junction and a desmosome and put it together? An intercalated disc. That's what that is. So the answer here is gap junctions and desmosomes. If you look back in, I don't know, was that chapter 4? We talked about, chapter 3, we talked about cell junctions. So the heart has a special disc, an intercalated disc, that enables each muscle fiber to connect with the other one electrically so that all the fibers together can act on the same impulse very, very, very fast. And they can share the same impulse. That's how the heart acts as a unit, right? You don't want one part of the heart doing a whole different thing, you know. It should all contract as a unit. Okay, lastly, skeletal muscle is definitely striated, voluntary control. And we're going to study what these light and dark bands mean. That's the next big thing. Because part of that structure that I talked about being the primary big thing in this chapter is including the sarcomere, which we'll get to. It explains all these dark bands that we see. So on the far left in this picture is your skeletal muscle. Okay. Also considering calling it a myofiber or a myocyte, same old thing. So again, if we think that all cells are round and just like we learned way back in the beginning, realize that a skeletal muscle cell looks like that, where we have nuclei, and if we dive inside of it, right, we're still going to have mitochondria, all the other stuff, right? We have endoplasmic reticulum in there. It just looks in a different orientation. It's a whole lot more organized. And the organization of the proteins that are found in skeletal muscle is why you get those bands. Think of them as, you know, army troops just lined up so perfectly. Whereas proteins and other types of cells are just floating around, right? Well, these proteins inside muscle, they're very lined up, very orderly. And that's what gives you the look of those stripes. Okay, so this is our grand organization. So starting from the top, skeletal muscle as a whole, made up of fascicles. Fascicles are made up of muscle cells. Muscle cells are made up of several myofibrils, and myofibrils are made up of myofilaments. And the myofilaments give us the organization of the, what we call the sarcomere. So know it from top to bottom, bottom to top. If we go the other way, myofilaments group together to become myofibrils. Myofibrils group together to become cells. Cells make fascicles. Fascicles make muscle. That's up and down. You got to know it backwards and forwards. If we look at the layers, the surroundings of them, the skeletal muscle as a whole, this is just a visual aid, of course, if hopefully you understand what's going on. As a whole, the muscle is surrounded by epimysium. The fascicle are each surrounded by perimysium. Each muscle cell has its own what? Sarcolemma, which is the cell membrane, which is then surrounded by the endomysium. Now, once you're inside the cell, what's the fluid inside of every cell called? Cytoplasm. But in the muscle, it's called sarcoplasm. So all of these internal filaments and fibrils inside the cell are being bathed. They're all surrounded by sarcoplasm, okay? So it's not necessarily a layer like a wrapping of an endomysium. It's more so just the environment around it, around the sarcoplasm. Okay. So seeing it another way, this is that same picture from before. There's our muscle fiber making up a fascicle, and each fascicle making up the whole muscle belly. So what comes next here on this blank? Myofibrils and then myofilaments. Right. So the two blanks in this slide indicate what's inside that muscle fiber right there poking out. And it's myofibrils and then myofilaments. And that's where we're about to dive into. Okay. There's your three layers. So just know peri is for the fascicle, epi is for the entire muscle, endo is around each muscle fiber. This picture is quite confusing because it's on a different scale. Right? Now consider this whole thing poking out is the muscle cell, Now, what is inside the muscle cell? Each of these units is a myofibril inside of that one muscle cell. The whole thing is a fascicle there. That's what kind of messes people up on this slide. So if you ignore this background here, pay attention to what's inside the muscle cell itself. These are myofibrils, and what's poking out of there?
Filaments.
Myofilaments. So that shows you how filaments become fibrils. Fibrils become the whole muscle. It's just a layer, one layer in to what we're seeing on the previous slide. So what's the surrounding that we see? This blue or purplish surrounding around the muscle cell? Sarcolemma. And that white connective tissue that's surrounding all these right here would be the endomysium. See, it's labeled endomysium? Right? Okay. Good. So I'm giving you several options. If you want to learn it this way, there's no picture on the test, of course. Right? So if this is good for you, then fine. Learn it that way, looking at the picture. But if this works better for you, then learn it this way. It's your choice, right? That maybe is easier, so you don't get confused. Because you're always safe with that. And I promise you there's a big old matching section with all this on there. So count on 15 points out of the exam automatically going here. Right? So you got 15. Then, do you know that striated is, is it skeletal and cardiac or smooth? Right, skeletal and cardiac. And since skeletal is striated, is it voluntary or involuntary? It's voluntary. That kind of stuff is easy. You should know that. Okay, moving on. All right, now here is where we get to the internal anatomy. Let's start here. We said that the muscle cell, is a long cylindrical cell with multiple nuclei, right? Now, each of these muscle cells is going to be surrounded by what? The cell membrane, which we call the sarcolemma. That sarcolemma will then be surrounded by a endomysium. So if we go back to our marker, the plastic that makes the marker, the coating around that marker, as an example, is the sarcolemma. My hand wrapping around the marker as a muscle fiber would be the endomysium. Okay? Independently. Now, if we consider that cell membrane now folding in upon itself into the cytoplasm and then back out again, right? Imagine the cell membrane folding in. Remember how the microvilli and the flagella and all that kind of stuff, the cilia, those are projections that go out? Imagine projections that come in, dipping into the cytoplasm and coming back out again, like a canal. That's going to give us the T-tubule. So we'll have these internal, what they call vertical invaginations of the sarcolemma, which go in, right? Because imagine now, if we excite that cell membrane with a stimulus, a new action potential coming from a nerve, we want that nerve impulse to dive deep into the cell. So that's how we get it there, is by the T-tubule. So imagine a blinking light on the sarcolemma, an electrical impulse, and now it falls into that tunnel, the T-tubule. It's called T because it transverses the cell, going down inside of it. So what are the fibers that it's going to ignite on the inside of the cell? What are those smallest filaments that we have? The myofilaments. That's what makes up the sarcomere. That's how we get that contraction, through the electrical impulse diving through the T-tubule. So sarcoplasm is nothing more than the cytoplasm of the cell. The reticulum here is nothing more than smooth ER. Parts of the smooth ER that we have to know is called the terminal cisterna. Anybody know what a cistern is? Like a collecting vessel, yeah, right? This is, in this sense, it's called a cistern because it collects calcium. So notice calcium is underlined. You should definitely link up in your brain calcium with terminal cisterna. It's like a bank of calcium, because we need calcium for the muscle contraction to happen. So where do we get calcium from? The terminal cisterna. Very, very, very good. All these associations with the bands, we're going to learn that in just a second. Just kind of hold off on that. Now, when we take two terminal cisterna, one on each side of a T-tubule, we get what's called a triad. So I'm going to show you a picture in just a second. But now you know that a triad is that one T-tubule, which is what? Part of the sarcolemma that dives in, right? Surrounded by two terminal cisterna, and we call it a triad. This is where all the action takes place, where the stimulus comes down to ignite those myofilaments. So I have a description here. Each terminal cisterna is going to release available calcium into the sarcoplasm. The T-tubule will carry that stimulus from the sarcolemma. And how did it get to the sarcolemma, the stimulus? From the neurotransmitter across that synapse. Right? So that's what we're going to see coming up, is the big story of the axon sending the impulse to the muscle cell receiving that impulse, igniting the sarcolemma, traveling down the T-tubule, mixing with calcium and a little bit of ATP, and then we get what? A contraction, finally, right? So that's what's, it's so many words just to describe that. This is our whole test. This. Just bending your finger, right? But we take 3 weeks to explain it all. It's amazing. And of course, what do we get out of the mitochondria? ATP. So the recipe for contraction is ATP, calcium, a threshold impulse, and what else? O2 and glucose. And that really is what gives you the ATP anyway. So getting all the recipe for a good contraction, you need adequate oxygen, adequate glucose so that you can make ATP. You need available calcium and a good impulse, a good threshold impulse. And that gives you a full muscle contraction. So realize by doing it that way, you realize the pathology. If you're without oxygen, or if you're without ATP, or if you're without glucose, or a threshold stimulus, any one of those will cancel out a contraction, right? Or a future contraction. Yeah, that's what happens when you see a running back running down the field and all of a sudden they get a horrible hamstring spasm. Because that hamstring, it might have just run out of O2. Maybe that guy didn't warm up. Or maybe he was up too late drinking the night before. All those kind of things can make a muscle go haywire. Okay, what we also see inside the muscle cell, stored glucose, which is called glycogen.
Like when the runner hits the wall.
What's that?
Like when the runner hits the wall.
Yeah. So we know muscle to be high storage of glucose. When you eat that bag of chips, right? Or whatever, and you don't burn all that sugar off, it gets stored as glycogen in the muscle and the liver. So the first 20 minutes of exercise, you're basically burning off the muscle that's stored, the glucose that's stored in muscle. Then you start burning fat, roughly at about 15-20 minutes. But the first part of exercise, you're burning off glycogen. Myoglobin is the red pigment that gives you the color of muscle there. That's the amount of oxygen that's stored in the muscle. That's why, if you would imagine, go back with me here, that kind of muscle that is high endurance, like leg muscle, is the leg of a chicken. Is it dark meat or white meat? Dark. That's the myoglobin that you're seeing. The dark color is the high level of myoglobin, which means it can store a lot of oxygen. That means it lasts longer. Legs don't give out. You ever try to chase a chicken? They don't really get tired, right? They run all over the place. I used to have 2 chickens. Oddly enough, when I grew up in New Orleans.
Like pets.
Yeah. I had to give them away when they got full grown, They got to be mean. They used to poop all over me. I'll never forget my dad said, "Okay, Mike, put one on each shoulder." I said, "Dad, I don't want to do that because they poop all the time." Sure enough, as soon as he clicked the picture, they both, pooped on each of my shoulders. Yeah, I'll never forget that. Speaking of poop, one of the first dates my wife and I had in New Orleans, we went to visit French Quarter, right? She spent all day trying to look her best, right? Which she did. But I'll never forget, she got pooped on by like 3 different pigeons, just one after the other. It's like they were chasing her. Poor girl. That's terrible, man. Of course, it's funny, but terrible. Myofibrils are just internal protein fibers inside the cell. So here it is. This is our muscle cell, the whole thing. So what are each of these cylindrical units? We have 1, 2, 3, 4, 5. What do we call each of those 5? Myofibrils. The picture is not labeled very easily. It's pointing to the little red dots, isn't it? Which is misleading. So maybe you can fix your picture. Each of the little red dots inside is a myofilament, but the whole unit is a myofibril because this whole thing together is one muscle fiber. So if the whole thing is muscle fiber, what's this sheath we got around it? This is the cell membrane. This is the sarcolemma. So that sarcolemma is surrounding the entire muscle cell. Inside the muscle cell, we have 1, 2, 3, 4, 5 myofibrils. Do you see them? Okay, the big cylindrical units are each myofibrils. And the little dots inside of them, the little red dots inside the myofibrils, those are the myofilaments. Okay, so here's where we use our anatomy we just learned. Here's our triad. The triad is what? A T-tubule, which is like a tunnel, a channel from the sarcolemma coming across, right? And there's our terminal cisterna on each side. So one terminal cisterna, another one in the blue or purple with a T-tubule in the middle. That gives you the triad. Can you see how if we excite one part of the sarcolemma with a stimulus, how that stimulus can travel down to the T-tubule and then make its way across all these fibers? And like fireworks, it ignites all those fibers as the impulse comes down. With the help of calcium and ATP. The oxygen comes with the ATP, actually. That's how you get the ATP. But you still have to have oxygen, yeah. Okay, so just using our anatomy. So next story is getting to know what all the world these lines mean. So the whole sarcomere is nothing more than the description of one Z line to the next. They call it a Z disc. Z line is just fine. It's the same old thing. So from one Z line to the other Z line, that dimension is considered one sarcomere. Okay, one sarcomere, which is composed of lots of myofilaments. It's a 3D, shape. There's above, below, coming at you, coming away from you, right? Lots of myofilaments in one sarcomere. Okay. So what we see is the band that's created all the way from this is the red filament here is a thick filament. The smaller blue filaments you see are the thin filaments. If we go the dimension from one thick all the way across, that's called the A band, right there. The area where there's no thick filament, see this region right here? That's called the I band. They get the I and the A. I stands for isotropic, which means light can pass through. A means anisotropic, which means light does not pass through. So when you look at skeletal muscle under microscopy, electron microscope, you see these light and dark bands. What is the light band? Where the I band is coming through, because no thick filaments are blocking it. The A band is that thick dark band where it is blocked by all the thick filaments. Anisotropic. A-N-I-S-O, and it looks like tropic, but it's pronounced tropic. Okay. So do know what the I band is. The I band represents the distance or the space between the thick filaments. From one thick filament to the next, right? Because there's no thick filament there. So the I band contains, this is what you really need to know. The I band contains what? What kind of filaments? Thin, which is also called the actin filament. Thin is actin, thick is myosin. The red filament is the myosin filament. So what kind of filaments do you see in the A band all the way across? You see thick and thin. Beautiful. What about this H zone? What kind of filaments are in the H zone going all the way up and down? Just thick. Because there's no thin filaments in that H zone, if you can see that. So that's the takeaway. You know that there's no picture, of course, on the tests, but you have to know the contents of each of these bands. And I'll say it one more time. The contents of the I band are actin filaments only. The contents of the H zone are thick filaments, or myosin filaments only. And the contents of the A band, think of A for and, if that helps, is thick and thin filaments in the A band. Okay, perfect. And what is a Z line? What does that represent? One sarcomere. It's from Z line to Z line. This is the craziest part of the chapter. I remember hating this stuff when I took it, and I never understood one word of it. It honestly takes just looking at it a couple of times. There's one section of the test where you're going to have to tell me what an I band is, an A band is, and I just told you what I need you to know. I band contains thin, H zone contains thick, A contains both. Think of it as this: I is a thin letter, H is a thick letter, and A stands for and. Maybe that helps. That just tells you what's contained in those areas. But this is really what's giving you the bands. So if we look out on this picture, here's all the bands. Here's the striations. The dark band is nothing more than the A band. The light band is considered the I band. What's in the I band? Thin filaments. That's how come we can see right through it. What's in the A band? Both filaments. That's why it appears dark when you shine light through the muscle. Great. Okay. So what's this peeling around it? Sarcolemma. This represents an entire muscle fiber. So what are all these little units inside the muscle fiber? There's probably a good 50 of them. Myofibrils, right? If we poke out whatever protein filaments are inside the myofibril, myofilaments. Beautiful. The thin filament is called actin. The thick filament is called myosin. Excellent. That's everything I just said is what you need to know. Right? That the entire muscle is surrounded by, the entire muscle fiber, surrounded by sarcolemma. It's made up of myofibrils. Each myofibril is made up of myofilaments. The thin filament is the actin. The thick filament is the myosin. So what we're going to see, the way these actin and myosin filaments play off each other, think of this, my arm, as a thin filament, right? The thick filament right here. Oh, let me do this. That way it'll be more dimensional. There we go. So now this stick is a thin filament. The thick filament's right here, my hand, my arm. The thick filament is going to rise up, grab on to the stick, the thin filament, and pull it. And that's what a contraction is. A contraction is nothing more than the myosin head being interested enough to come into position, bind to the actin filament, and do what? Pull. What fuels that to happen? ATP. What helps this to bind? Calcium. So remember that calcium from the terminal cisterna? How we depend on it being there? That's where calcium is used. In order for the myosin head to bind to the actin, you have to have calcium around. Otherwise, there's no binding. Right? Once you get a binding, Remember how when you bind something, there's always a changing of shape? Right. The calcium binding to troponin, which we're about to get to, is what makes that changing of the shape. That's what helps the binding of the myosin to the head, to the actin filament, and then you get ATP fueling that power strip. So that's what's about to be explained coming up in about 3 different ways. So hopefully you get it. But this is just a summary of what we said about all the units there on the sarcomere. So if you're confused about what I'm holding you to, it's all there, okay? This is what you need to know. The sarcomere can be very mysterious. It can put a drag on the whole chapter, really. But it comes down to, do you know this? Right there on the board. And do you know that the Z-line separates each sarcomere? And it's what divides the I band right in two. Okay, one more look at it now. If we look at that myofibril poking out, now we see the full, we got 3 different sarcomeres going on here. Right? The light and dark bands are created by the I band and the A band. Okay. Now here's a better look. On the bottom here, look at all the, this is myosin. Right? This is the whole thick filament. And the thin filament here. Thin in blue, thick in red. Why is it called thick? Because it's thick. It's got lots of myosin protein units. You see these little nubs poking up on the red thick filament? Each one of those little nubs is a head, a myosin head. Notice it's 3-dimensional. So we got heads poking up, poking down, poking out towards you, and poking out away from you. Because this is all a cylindrical unit. So it's not flat like we see it in the picture. It's cylindrical. So how do we get these heads to come up to reach the actin?
ATP.
ATP helps to energize those heads to come up. Right? So that's what we're about to see. But I just wanted you to get the overall anatomy going. So what these are considered to be is regulatory proteins. So that's what we're going to see. Structural, contractile, and regulatory. The structural proteins help keep the structure of the sarcomere together. The contractile make the contraction happen, which is the actin and myosin. The stick that I showed you earlier, right? That was a contractile protein. The regulatory proteins regulate if the contraction happens or not. So let me go back. Notice these little coils there. That's a structural protein. And what's it doing? It's holding the myosin heads in place structurally so they don't just fall apart. This right here. This is tighten. The myomecin represents the proteins that line up the filaments like this, holding them in place. So the M line is that myomecin structural protein. The contractile proteins are the actin and myosin themselves, and the regulatory proteins are going to be the ones that regulate whether we get a contraction or not. And that's what's coming up, the troponin and tropomyosin. So I'd rather you just focus on the contractile proteins, actin and myosin, on this slide here. Please know those. Those are the myofilaments that we've been spending the most time on. We've just recognized these as structural proteins. They help keep the structure of the sarcomere together. The regulatory is going to help us with the story of how we get a contraction. Okay. So here we go. Here's our thick filament. There's our myosin molecule making up the thick filament. So the thick filament is the whole thing. The myosin protein makes up one of all of these, right? So it has a head and a tail. The heads are what come up to bind to the actin. So here's our actin molecule. Right? Our thin filament is made of actin proteins swirling around each other. So check it out. Notice the dark circle on each of these. That's the binding site that we want the myosin head to bind to, but they're covered up. What's covering them up? Tropomyosin is that orange protein covering up all of our nice little binding spots. So we need tropomyosin to move, don't we? So that our myosin head can come up and bind to each one of those little dark circles. So how do we get tropomyosin to move? We have to get troponin to tell it to move. So here's the story, okay. Number 1, calcium. Number 2, calcium binds to troponin. Okay, so I'll start again. Number 1, we have to have available calcium, and where is it coming from? Terminal cisterna, right? Smooth ER. Part of the triad. So link those, because it could be test question any which way. Anyway, available calcium is going to bind to troponin. With that being bound, troponin changes shape and pulls tropomyosin off. So again, number 1, available calcium. Number 2, calcium binds to troponin. Number 3, troponin pulls tropomyosin off of the binding site. When tropomyosin is pulled off the binding site, now the myosin head is going to come up and bind to what? That available binding site, right? Okay, so here we go. And that tells you everything I just said. So this is what regulates the contraction happening. Are these molecules troponin and tropomyosin. So it's the actin filaments that are being pulled by the myosin heads. Each one of these myosin heads comes up, attaches to the actin molecule, the binding site, and then pulls the actin. So imagine in this picture, imagine these actin filaments coming together, right, toward the middle, and that's what creates the overall contraction. Myosin doesn't move, only the actin moves. The head goes into a cocked position and back, but it's only the actin filament that actually slides. They call that the sliding filament theory. I'm not sure why it's called a theory, because it does exist. So what you're going to see is this band right here, the H zone, disappears because the thin filaments come together. So what do we need for this to happen? ATP and calcium. So what does ATP do? It's all there. Energizes the myosin head to attach to the actin for the power stroke. Now, once the power stroke has been completed, what do we need to happen? A detachment. We need to be able to let go of that binding, right? And what enables us to let go is another ATP molecule. Helps us detach. So that's why we get rigor mortis. If we die, then our muscles may contract with the last little bit of calcium available and ATP. But since there's no new ATP flooding in, because you're dead, then you stay contracted, right? Until the proteins break down, then you naturally just kind of, fade away. Okay, so ATP is used twice. Once for the power stroke, once for the detachment. Right? And then if you want to start the cycle again, then a new ATP will come in again, right? But 2 to start off. Calcium, where is calcium used? It helps bind to troponin, and what does that do? Helps to pull tropomyosin off. So this is the hardest part of the chapter, I promise, is this little sticky area here. Calcium binds to troponin, which then pulls tropomyosin off of the myosin binding site. Where was calcium used initially, besides in the muscle? Where have we talked about calcium before? Before we even get into the muscle. Well, bones, yes, but I'm talking about for the nerves. for the neurotransmitter to be released? Remember we said calcium had to be influxed in for the neurotransmitter to be released? So now we're rewinding if we can access those parts of our brain even. Do you feel a little like bruised and shaken up? All this stuff we're shoving in your brain? Well, save room for AMP2. There's more to come. It's not easy, though. I mean, I find this fun because, I know it. But I remember how it feels to not know it, and, to try to put all this together is a big thing. This picture here is just showing you what a relaxed versus a contracted sarcomere looks like. What's the difference? The actin filaments are overlapped here in a contracted sarcomere. Okay. This is an electron microscope picture of a neuron approaching a muscle fiber. Isn't that cool? Now, although we see it touching, if we magnified that junction even further, you'd see a space. That our good old synapse, right? Okay. And that's just supposed to be sarcolemma right there. So now we're pulling together the whole story. We're going to start from the nerve impulse and see how it travels into the muscle. Maybe this will help to see it all in one place. Okay, remember our good story? You're like, oh no, not again. Well, again, we have to start at resting potential. And what comes next after resting? Depolarization, which means an adequate stimulus has come through. That stimulus, it creates a depolarization locally on the cell membrane of the axon. We're back at the axon now, right? We want that charge to come down the axon all the way to the axon terminal, and that's where we're headed next. As we move along, it's going to repolarize, of course, wherever it came from. Any questions on that? As we depolarize down the cell of the axon, the area previous will repolarize, right? That's all we're saying. Okay. So now we get to the end of the axon, right here, the axon terminal. So everybody raise their hand. Everybody knows what the end of an axon is called? Axon terminal. Beautiful. What's at the axon terminal? Holy cow. Vesicles with neurotransmitter inside. What's our neurotransmitter of choice for skeletal muscle? Is it labeled somewhere? Acetylcholine. Acetylcholine is our neurotransmitter that we use for skeletal muscle. Acetylcholine. ACH for short. So, how do we get that acetylcholine in the green to be released from the axon terminal? It is exocytosis, very good, but we need an influx of calcium, right, to tell those vesicles to send them out. Without calcium, the vesicles don't hit that cell membrane and let them out. So calcium, we call this event calcium dependent. The fact that we need it so the neurotransmitter is released. Now that the neurotransmitter is released, we jump to this other picture on the far right. What's happening here? There's a binding of acetylcholine on a sodium channel, right? If it's a good binding, the sodium channel opens, and what happens? Sodium rushes in. Great. Okay, so here's the whole story. Now that we depolarize the cell membrane of the muscle, because this is a muscle now, right? Now that we depolarize the cell membrane of the muscle, it's called the sarcolemma. We carry that charge down the T-tubule. When that stimulus is coming down the T-tubule, calcium is released into the sarcoplasm. When calcium is released into the sarcoplasm, what happens? What does calcium bind to? Troponin, right? What does troponin do when it's bound to calcium? Changes shape, pulls the tropomyosin off, then the myosin head connects, binds, and pulls, which is the power stroke fueled by ATP. So this is a huge headache of a story. The good news is I have a handout.
Yes.
that shows you the 10 steps. A handy handout, that's right. With everything in order. And are you going to have 50 questions on this? No. It's going to be a few questions. So even if you don't get it, it's not going to kill your test grade. It's just one thing among many. But it is a big story. Now, once that ATP fuels that power stroke, realize we need a different ATP to do what? To detach. And that's the end of the story. So it's a good story. And there's all the bullets, right? So you can read all that. And there's all the graphics that go with it. So let's do it one more time. Motor neuron releases ACH, which is calcium dependent. Electrical impulse is generated on the muscle cell membrane, which we call the sarcolemma. It dives down the T-tubule. The terminal cisterna releases calcium. So here's our two uses of calcium right there. Test question. Ding, 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 ding. Right? Yes. What's next?
Summer.
Yeah, summer. Let's just fast forward to summer. I hear you. The calcium binds to troponin. Troponin changes shape, pulls tropomyosin off. An energized myosin head. What do we use to energize the head? ATP. Right? Now the myosin head gives the power stroke using that same ATP. And then a new ATP molecule will help it detach. So here's your two uses of ATP. And on the previous slide is your two uses of calcium. You should know that. That sounds like a great test question. And not only that, you have my permission to share this with the world. Yeah, it's a great story. This is the only way I can, I find it hilarious that I'm up here teaching this because I never, ever understood this in school, ever. Because my teachers are just, I don't know what they were talking about, right? I was reading something else in the back. I really didn't care. Until I met that one teacher that started to make sense, right? And which inspired me to want to know it because it's just that interesting. So hopefully it does inspire you. Go back where?
Just the previous one.
Okay. That one? Yeah. Yep. Because it is learnable. It sounds hard because there's a lot of steps, but that doesn't mean it's not learnable. Okay. So a muscle contraction can continue as long as calcium and ATP are available. Here's just some graphics to show the binding, the pulling, the detachment. Just pictures out of the book. It helps if you read the caption and understand the story, the more exposure to it, the better. Okay, we talked about rigor mortis. That's just the fact that new ATP is not available. So you stay contracted.
Is Bubba going to get rigor mortis?
I should have that, and I don't have him decomposing on the test. I might think of something. Good old Bubba. All right. The rest of this PowerPoint has to do with some of the, let me see where, I think it's going towards the special features of muscle. I want to take this time, yeah, we've only got about a good 15 slides left or so. It's not that bad. I want you guys to work on this case study a little bit because you'll find it fun.